Hello, my name is Dr. Nizar Hanzik and I'm professor of biology at Oakton College. In this lecture presentation, we will go over the steps of humorally mediated branch of adaptive immunity. For that purpose, I've developed an immune system manipulative. So we will now move to the whiteboard, or in this case, to my door, so we can go over those steps. So here we have an antigen-presenting cell, such as a macrophage. Macrophage, of course, is a phagocytic cell. It has detected and engulfed a foreign pathogen, such as a bacterial cell, through a process known as phagocytosis. This foreign pathogen, meaning bacterial cell, is now trapped within the macrophage within a vesicle known as phagosome. This phagosome will consequently merge with lysosome, such that the lysosome will release hydrolytic digestive enzymes into the phagosome. These digestive enzymes will consequently break down this bacterial cell into smaller fragments. Some of these smaller bacterial fragments will then be displayed on the surface of this macrophage to specific receptors. One such specific receptor is MHC type 2 receptor, major histocompatibility complex type 2. Coupled to this receptor is a fragment of the bacterial cell, which will then act as a foreign antigen, Ag. Now, this macrophage is ready to present this foreign antigen to another immune cell, principally helper T lymphocyte, in the process, of course, activating the helper T lymphocyte. So here we have the helper T cell, which on its surface contains T cell receptor, TCR. Notice that this receptor is complementary in shape to the shape of this foreign bacterial antigen presented by the macrophage. Notice how these pieces are fitting together like Legos. This physical connection between the macrophage and the helper T cell is further reinforced by a protein marker known as the CD4 protein. This antigen-presenting cell will activate this helper T cell through two mechanisms. The first mechanism is physical connection between the macrophage and the helper T cell through these surface receptors, which are complementary in shape. Remember, this connection is reinforced, stabilized by CD4 protein marker. The second mechanism is through the release of various chemicals known as cytokines. These chemicals are released by the antigen-presenting cell and they will bind to the helper T cell, in the process stimulating such cell to become activated. One such potent cytokine is interleukin type 1, IL-1. This chemical, interleukin-1, is released by the macrophage. It will bind to the helper T cell and it will stimulate it to activate and thus to rapidly divide, forming many copies of itself, many daughter cells. In other words, this antigen-presenting cell is saying to this helper T cell, hey, I have captured a foreign invader. This is what it looks like. Now go forth and spread the word, spread the image of this foreign invader to other immune cells within the body, such as B lymphocytes, in the process activating these cells to rapidly divide and thus to mount, to generate a very strong defense against this foreign invader. So now this helper T cell will activate another immune cell, in this case B lymphocyte. Here we have our B lymphocyte, right here. Similar to the macrophage, the B cell, or the B lymphocyte, is also a phagocytic cell. It has detected and engulfed this foreign invader, the bacteria, through a process known as phagocytosis. Now, this B cell will display foreign antigens on its surface coupled to the surface receptors. The name of such receptor is MHC2, Major Histocompatibility Complex Type 2. Coupled to this surface receptor is a fragment of the bacterial cell which will act as a foreign antigen known as AG. The helper T cell, of course, on its surface has a T cell receptor, TCR. Notice that this TCR, this receptor, on the surface of the helper T cell is complementary in shape to the shape of this foreign antigen, bacterial antigen AG. This physical connection between the helper T cell and the B cell is further reinforced through a protein marker known as the CD4. This helper T cell will stimulate this B cell to activate and to rapidly divide through two mechanisms. One of such mechanisms is physical connection between the helper T cell and the B cell via these surface receptors which are complementary in shape. This connection, once again, is reinforced, stabilized by a protein marker known as the CD4. The second mechanism of stimulation involves the release of various chemical cytokines from the helper T cell. One such potent cytokine is interleukin 2, IL-2. This interleukin 2 is released by the helper T cell. It will bind to the B cell and it will stimulate it once again to activate and to rapidly divide, thus generating many daughter cells, copies of this B lymphocyte. Consequently, some of these daughter cells will differentiate into effector cells, plasma cells, and others will differentiate into memory B cells. Plasma cells are short-lived soldier cells. These cells are responsible for the production of antibodies. These antibodies will consequently disable this foreign pathogen through a number of mechanisms that we will discuss shortly. On the other hand, the memory B cell is a long-lived immune cell. It will survive within the body for decades, if not for a lifetime, such that should the same individual be exposed to this same bacteria many years from initial exposure, these memory B cells are now present within the body in great quantities, numbers. They will quickly mount a strong defense against this bacteria, disabling it, destroying it, before this bacteria can wreak havoc within the body, before the bacteria can cause disease. And as such, this person, this individual is now said to be immune to that bacteria. The plasma cells, of course, will produce goodly amounts of antibodies, which will then consequently disable this pathogen through a number of mechanisms. These include neutralization, agglutination, opsonization, and cytolysis. In neutralization, these antibodies will neutralize this foreign pathogen. So for instance, these antibodies may bind to the surface receptors located on the surface of the virus, thus preventing the virus from entering the host cell. Remember, the virus must enter the host cell in order to replicate itself. Additionally, these antibodies can bind to the toxins, such as those produced by the poisonous snakes, thus disabling, neutralizing these toxins before they can damage the tissues of the body. In the second mechanism, known as agglutination, these antibodies will cause the bacterial cells to agglutinate, to clump together. This will then greatly increase the rate of phagocytosis of these foreign bacterial cells by big eaters, macrophages. Thirdly, these antibodies will act as opsonins. Certain bacterial cells will tend to hide their foreign protein markers, meaning foreign antigens on its surface, under a thick polysaccharide capsule in order to evade detection by the macrophages. So these antibodies will attach to this polysaccharide capsule, making these bacteria now visible, more appetizing if you will, to these big eaters, macrophages, such that the macrophage will detect it as foreign, engulf it, and destroy it through a process known as phagocytosis. And lastly, these antibodies will destroy foreign pathogens through a process known as cytolysis. In this process, antibodies will activate the complement system within the blood, which will then result in the generation of MAC, membrane attack complex. This membrane attack complex will generate holes within the cell membrane of the bacteria. Through these holes, ions will enter, which will then osmotically pull the water into the bacteria. The bacteria will consequently swell up and lyse rupture open. Not a nice way to go. All right, we will finish our presentation at this point here. Thank you for watching.